Yeah, great. Thanks. It's a great pleasure to participate in this meeting and present our small talk about our research. Modern cities harbor a high diversity of plants, and urban floras are significantly different from natural floras, especially when considering the proportion of alien species found in cities. Taxonomic diversity is well studied for Russian cities, but we cannot say so for other diversity facets and phylogenetic diversity in particular. My talk is devoted to the investigation of phylogenetic diversity of urban floras of 4 cities located in the Central Urals region of Russia. The Urals is a geographical region located around the Ural Mountains. It extends approximately from the Arctic Ocean to the bend of Ural River. The map shows the administrative partition of Russia into 7 federal districts. So you can see that Ural region in the central part of Russia on the border between Europe and Asia. We investigated floras of 4 cities in the Sverdlovsk region: Krasnoufimsk, Yekaterinburg, Kamensk-Uralsky, and Turinsk. Yekaterinburg is a regional capital. It has a population of almost 1.5 million people. 3 other cities are smaller, with population from 17,000 people to 170,000 people. The urban flora was regarded as a flora of the urban development zone with adjacent areas of forest parks or city green zones. The species lists were obtained from the Field Service, covering every type of habitat from herbarium collections. We distinguished 4 fractions of species in the urban floras. Native species were classified as indigenous or epiphytic, based on whether they exhibited expanded ranges because of their association with disturbed habitat. The indigenous plants are those that generally avoid anthropogenic habitats, while epiphyte plants are associated with anthropogenic habitat. Alien species were divided into 2 groups based on their residence time: archaeophytes and neophytes. Archaeophytes appeared to the Central Urals before the arrival of the Russian population in the 18th century and earlier. Neophytes appeared in the Central Urals after this date, 1800. To quantify phylogenetic diversity, we used dated phylogenetic tree published by Amy Zanne and colleagues. From 1,072 plant species recorded in our species lists, 806 were found in the phylogenetic tree, and the rest of 266 species were added as polytomies to the corresponding genera or families. We calculated 4 indices for each urban flora and for all fractions of species. First, species richness, this is most basic quantity, and 2 indices of phylogenetic diversity. Fates index, which is a sum of all branch lengths of the phylogenetic tree connecting all species of a given flora, and the second measure of the phylogenetic diversity, this is the mean pairwise distance. And we also performed a null model analysis, which compares the phylogenetic diversity in each fraction to null expectations generated from randomizations of species membership of each fraction. The final quantity is called standardized effect size. Here you can see the overview of urban floras of 4 cities in the Central Urals. Floras are dominated by native plants, which constitute about 67% of species on average. Among native plants, indigenous fraction is larger than epiphyte fraction. Alien plants account for about 30% of urban floras, and they are dominated by neophyte fraction. Phylogenetic diversity assessed by the Faith Index was higher in the urban flora of Yekaterinburg and lower in the flora of Turinsk. This was expected because Faith Index is highly correlated to species richness. You can see the perfect ordering in the same way. That's why we applied another metric of phylogenetic diversity, mean phylogenetic distance, which is independent of species richness, at least theoretically. This index shows that all 4 urban floras are characterized by comparable diversity. The most interesting results in our study were obtained with null model analysis. We adopted approach developed by Campbell-Webb, who proposed to calculate mean phylogenetic distance, MPD, and compare it with the distribution in the null model. The simplest null model is a random set of species from a given pool. In our study, we regarded each urban flora as a pool and then randomized species identity in fractions of indigenous plants, epiphytes, archaeophytes, and neophytes. Then we calculated normalized value of MPD. Here, MPD obs, this is observed MPD. This is a value calculated on the basis of empirical data. MPD null and SD MPD null. This mean and standard deviation, respectively, of the MPD values calculated for random sets of species. Let's consider a specific example. Here is a histogram of the distribution of the mean phylogenetic distance in the null model for the fraction of native species in Yekaterinburg flora. The empirical value is approximately 332 million people. At the same time, it is clear that random sets of species have a phylogenetic distance that is significantly smaller than empirical value. Then standardized effect size will be positive. This situation is called phylogenetic overdispersion. The standard interpretation for this case is that the structure of the community is determined by biotic interactions that lead to competitive exclusion of closely related species. And here is the counterexample, fraction of alien species in Yekaterinburg flora. The empirical value of MPD is smaller than majority of the distribution in the null model. In this case, the standardized effect size will be negative. This situation is called phylogenetic clustering. The relatedness is higher than in the null model, and standard interpretation for this case is that the structure of the community is determined by environmental filtering, which allow only species with a certain set of traits to exist in the community, including closely related species. Our results show that in all 4 urban floras, the mean pairwise distance of native species exceeded the random expectation for MPD. In this case, standardized effect size is positive. This indicates phylogenetic overdispersion in this fraction. On the contrary, for a group of alien species, standardized effect size was negative, and this fraction exhibited substantial phylogenetic clustering. Interestingly, the 2 native plant fractions behaved in fundamentally different ways. The phylogenetic overdispersion of native plants was caused by indigenous species, while clustering was apparent for epiphyte species. Both alien fractions exhibited phylogenetic clustering. The effect phylogenetic overdispersion in the fraction of indigenous species could be explained with information about habitat preferences and geographical distribution of species. Indigenous species are adapted to a wide range of conditions and prefer various semi-natural environments. Here we have forest, meadow, steppe, and rocky species, as well as aquatic swamp and halophytic species. On the contrast, The strong phylogenetic clustering among alien species and epiphytes implies that these closely related species share a few suits of ecological traits that allow them to adapt to certain environmental conditions and coexist in similar environments, and are the outcome of phylogenetic conservatism of niches. We believe that the phylogenetic clustering of alien species and epiphytes is explained by environmental filtering, where the conditions of urban areas determine a complex of functional traits, and the owners of which will be adapted to local urban conditions. So here are my final points. Urban flora is characterized by a high richness of alien species. Moreover, their phylogenetic diversity is inferior in comparison to the native species. This reflects the high taxonomic, geographic, and ecological differentiation of native species and their ability to survive in broader ecological conditions. The group of alien species is not a random set of species. It includes closely related species with similar functional traits adapted to an urban environment. Thus, the high species richness of the urban flora of the Ural cities, as well as European ones, is provided mainly by closely related species that are functionally similar and adapted to an urban environment. Thanks for your kind attention.